I'd like to welcome everyone to today's Wednesday talk with Janet Rossant. We're all very enthusiastic to hear what Janet has to say. What I'm going to do at this moment is to mute everyone, and that'll mean that Bill Logan and Janet, you'll have to unmute yourself when you have time to speak. I think people may want to use the speaker view during the talk itself. At the end of the talk, we'll take a short break for coffee, tea, or whatever else is required, and then we'll come back for question and answers. So at this moment, I would like to turn things over to Bill Logan to introduce Janet.
Hi, Janet. Pleasure to introduce you. Dr. Janet Rossant is a senior scientist in the Developmental and Stem Cell Biology program and Emeritus Chief of Research at the Hospital for Sick Children in Toronto. And most recently has become president of the Gairdner Foundation and scientific director. She is also university professor in the Department of Molecular Genetics at the university. She trained at the universities of Oxford and Cambridge, United Kingdom, before coming to Canada in 1977. She is an internationally recognized developmental and stem cell biologist, exploring the biology of the early embryo and its stem cells, and their applications to understanding and treating human disease. She has pioneered many technologies for manipulating the mouse genome, and her interest in the early embryo have led to the discovery of a novel placental stem cell type, the trophoblast stem cell. Dr. Rossant has been recognized many times for her contributions in science with many awards, including the International Society for Transgenic Technologies Prize, the Ross G. Harrison Medal Lifetime Achievement Award from the International Society of Developmental Biologists, the Killam Prize for Health Sciences, the March of Dimes Prize in Developmental Biology, the Conklin Medal from the Society for Developmental Biology, and the Canadian Institutes of Health Michael Smith Prize in Health Research, which is Canada's most prestigious health research award. She is a fellow of both the Royal Societies of London and Canada and is a foreign associate of the U.S. National Academy of Science. She received the 2015 Canada Gairdner-Whitman Award, the first woman to do so. She is a companion of the Order of Canada. Dr. Rossant is actively involved in the international development and stem cell biology communities and has contributed to the scientific and ethical discussions on public issues related to stem cell research. She chaired the working group of the Canadian Institutes of Health Research on Stem Cell Research, which came up with guidelines for their funded research in this area. Janet, I'm so pleased and honored to introduce you and to thank you on behalf of Senior College for speaking to us today. And your topic is stem cells and gene editing, the science and the ethical debates. Thank you.
Well, thank you very much, Bill, for that kind introduction. And it's my pleasure to speak to everybody today. And the title of my talk says it all. We are going to talk about the science. We're going to talk about the ethical and the policy implications around stem cells and gene editing. So I'm going to share my screen. Here we go. And throw that up on full screen. And there we go. Everybody happy? We can see that. So stem cells and gene editing, the science and the ethical debates. So just to sort of summarize what we're going to be talking about today, this is the sort of overview of my talk. I'm going to talk about stem cells. I'm going to help describe what is a stem cell. I'm going to talk about very special stem cells, pluripotent stem cells, their origins and their potential, where we stand on stem cell therapy today, moving to the clinic, and then where we shouldn't be standing, which is the unethical use of stem cells, what we call stem cell tourism. And then I'm going to switch to gene editing, which is another extraordinarily important breakthrough in science that has the potential to treat and prevent genetic disease and also brings ethical issues. So we'll be touching on the science and the ethics as we go through. So what is a stem cell? A stem cell is a very special cell. It can divide and what we call self-renew. It can copy itself and make another stem cell, or it can actually differentiate into different cell types. So it has an option. It can divide and self-renew, make more stem cells, or it can divide and differentiate. And we know that there are stem cells in many different cell tissues of our body that have this capacity to differentiate into muscle, to nerve, to blood. And in fact, the stem cell discoveries go back to, sorry, just having difficulty advancing, here we go. Stem cell discoveries is really a Canadian discovery. It's a Canadian legacy. The first people to describe stem cells are the gentlemen here, Jim Till and Bun McCullough. And in the 1960s in Toronto at the what is now the Ontario Cancer Institute and the University of Toronto, these gentlemen described for the first time that in our blood, in our bone marrow, there are cells that are stem cells that make all the cell types of the blood. And those cells can reproduce. That's how we manage to keep making blood every day. And in fact, you can't read this particular picture, and I don't intend you to do, but this is actually shows you that stem cells and stem cell discovery has been a continuous Canadian advance. So stem cell science is really one of the areas where Canada excels. And beginning with Till and McCullough. And it's a very nice opportunity for anyone who's a stem cell scientist from around the world, they can go and meet Till and McCullough. They can go and sit on a stool and have a conversation with the statues of Jim Till and Bun McCullough outside of the Mars building here in Toronto. And here, in fact, of course, is Jim Till having a conversation with himself in 2017. So this is a very nice part of being a Canadian stem cell scientist, is to be able to actually meet your Canadian stem cell heroes. But when we talk about blood stem cells, the ones that were first derived and described by Till and McCullough, since that time, we've gone on to show that there are many different kinds of stem cells. And perhaps the most exciting stem cells are the cells that have the potential not just to make one cell type like blood or muscle or nerve or liver, but cells that can make all the cell types of the body. And this is where my research and research of a lot of other people comes into play. My research works on mouse embryos. And many years ago, we showed that in the mouse blastocyst, which is the stage of development shown here, it's the size of a speck of dust. It's got about 100 cells. And you can see there are pink cells and green cells. The pink cells, those cells in the middle, we call epiblast. And experimentally, we were able to show that those epiblast cells are pluripotent. That is to say, those little group of cells there make all the cell types of the fetus and the resulting mouse. And if this were a human blastocyst, the same thing would apply. The outside cells, the trophoblasts, make the placenta, which is necessary for the embryo to survive. And the green cells on the surface of the pink cells make another membrane, the yolk sac. So the concept that there are pluripotent cells in the early embryo that make every cell type was very exciting for me and for many other people, because what if we could capture that cell state? What if we could not just let this embryo go further, but capture that cell state and maintain these as pluripotent stem cells? And so this was the discovery that took place in 1981. Two separate groups, Martin Evans in Cambridge and Gail Martin in San Francisco, were able to take that little mouse blastocyst, and from the inner cell mass, from that epiblast, they were able to grow cell lines that they called embryonic stem cells that would grow indefinitely in culture, divide, 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 make endless copies of themselves. And yet, if you change the culture conditions, they would differentiate into multiple cell types. Nobel Prize went to Martin Evans in 2007 for this work, combined with work from Mario Capecchi and Oliver Smithies, who were actually the first to show how you could genetically modify mice using embryonic stem cells, which you might bear in mind when I come back to talk about gene editing later. So those ES cells then that they derived, they showed they could make lots of cell types in culture, but were they really truly pluripotent the way we had shown that the epiblast cells in the embryo were? Well, the best test of that would be to ask whether those cells that were growing in a petri dish could make a mouse. And so that's my favorite experiment of all time. And it's an experiment I did with my colleague Andras Nagy when we were at the Lunenfeld Tannenbaum Research Institute here in Toronto. And what we did was do a little bit of a trickery. We knew that the embryonic stem cells should be able to make the entire fetus and the entire mouse. But of course, they needed that placenta for support. And the embryonic stem cells would not be expected to make the placenta. And we knew that if we made embryos, normal embryos, and doubled up their DNA, make them tetraploid, then those embryos were pretty good at making placenta, but couldn't make the fetus itself. So if you combine the two, it's a sort of complementation system. You put the purple ES cells with the green tetraploid embryo, then what happens is you get a purple fetus and a green placenta. So that was pretty exciting. What was more exciting was when we were able to show that you could make mice this way. So you could take embryonic stem cells, aggregate them with tetraploid embryos, and make living mice that are entirely derived from the cells that were grown in a petri dish. And they're glowing green here because the ES cells were carrying a green fluorescent protein gene. So every cell is GFP positive. These are perfectly normal babies. They grow up into perfectly normal mice. So that experiment then really demonstrated that pluripotent stem cells, which can be derived and grown exponentially into millions and millions of cells, could be an incredible source of cells to replace every tissue in the body. But obviously not just in mice. The question is, could you translate that into humans? And can you make human embryonic stem cells, not to make human embryos, but to make a source of cells for cell-based repair? If mouse ES cells can make a whole mouse, what would be the power of human ES cells? So it took quite a while from 1981, when mouse ES cells were first generated, for Jamie Thomson in 1998 to first show that he could derive human embryonic stem cells, which looked very similar to what we had been able to show in the mouse. In the mouse were embryonic stem cells and could make a mouse. He did it by taking human blastocysts, isolating those epiblast cells, and getting these colonies of self-renewing cells. Those cells, over the years since, people have been able to take those embryonic stem cells in humans, and in the petri dish, by changing the culture conditions, you can generate multiple different cell types of the human body by differentiating them down different pathways. Cell types that could be used for potential treatments for serious degenerative diseases of the skin, of the brain, of the lung, of the kidney, of the muscle, many different diseases, as we'll see in a minute. So that was very exciting. Human ES cells, this pluripotent capacity of embryonic stem cells, really opened up new vistas in regenerative medicine. And then it became very controversial at that time. And the reason were the ethical concerns about not so much the cells themselves and their use for clinical medicine, but the source of these cells, that they came from embryos. And so the source of the embryonic stem cells were indeed human embryos. They were embryos that came from in vitro fertilization programs, where when you undergo IVF, there are usually too many embryos generated, more than are needed for reproductive purposes. And most of those embryos end up being discarded. So with informed consent, those embryos can be donated for research. And in this case, that research could allow the generation of human embryonic stem cells. But obviously, in certain religious backgrounds and in certain pro-life environments, any work with human embryos is considered unacceptable and unethical. And still today, the legislative restrictions on the use of human embryos in research varies considerably around the world. It is possible here in Canada. You can ask me questions about that later if you like. But in countries like Germany, for example, there's still no research with human embryos is allowed at all. So this is obviously, it was important. The cell type, the cell lines have an incredible potential, but it was obviously controversial. that led people to ask, well, do we have to use embryos to get pluripotent stem cells? Could you make the same cells with the same potential from a more accessible source, in perhaps a patient-specific manner? Maybe we could make your own stem cells for treating your own disease. And that really came about. There's a couple of game changers I'm going to talk about today, and this is certainly one of them. This was the discovery of iPS cells, or induced pluripotent stem cells. Shinya Yamanaka, a scientist in Japan, was the first to show that he could actually take skin cells anywhere from your body, a little bit of skin, bit of just easy biopsy, and in those cells express some of the genes that we and others had shown were important for making those early pluripotent cells. And by doing that, you actually turn back the clock. Expressing those early embryo genes essentially reprograms the skin cells to behave like embryonic stem cells. They become induced pluripotent stem cells. He showed this first in the mouse and then very rapidly thereafter in humans. And this changed everything. It certainly changed the ethical concerns, but it also opened up another lot of potential, because it does mean that you can make personalized stem cells. We can make stem cells from anybody. Any of you can donate your cells and you can have your own iPS cells. So this opens up the potential in the future for rejection-free transplantation from those cells. But it also means that we can model human disease in the petri dish, because you can take cells from people who have genetic diseases and study that disease from the stem cells in the petri dish and develop new drug screening and therapeutics. So it's really, really important. Yamanaka received a Gardner Award. Here's myself with my husband, with Shinya and his wife in 2009 when Shinya received the Gardner Award. And very shortly thereafter, in 2012, he received the Nobel Prize for Medicine for this work, along with John Gurdon, who was actually my undergraduate teacher when I was at Oxford, who was the first person to clone frogs, and therefore was the first person to show that you could actually turn back the clock and restart development by reprogramming the DNA. So pluripotent embryonic stem cells, or pluripotent iPS cells, are an incredibly important source of cells because they can make every cell type, and they have the potential then to treat serious degenerative diseases. But this is all very well. I'm making it sound like it's easy. And there certainly has been a lot of hype over the years about pluripotent stem cells and how fast we can move to the clinic. And I just want to give you an update on where we stand today and tell you that, in fact, finally, we are, I believe, close to moving some of the therapies from pluripotent stem cells into clinical practice. And so here are the ones that are currently moving towards the clinic. Spinal cord injury from human embryonic stem cells, not really repairing the nerves, but remyelinating the damaged nerves and helping alleviate damage. There are trials underway using that approach. Macular degeneration, this is actually a very important one, where there are several trials that are looking quite promising, where you remake the pigmented epithelium of the eye from embryonic stem cells, and you can transplant that back in and restore sight. Parkinson's, where people lose dopamine-producing neurons. There's a couple of big studies. The trials are underway in Japan, and there's an international consortium very close to trials in both Europe and the US and Canada, using dopamine-producing neurons from embryonic stem cells. Diabetes, type 1 diabetes. In Canada, we have the Edmonton Protocol. We know that you can transplant islets into patients with type 1 diabetes and take them off insulin. But there aren't enough donors to get islets that way. So the idea is to make islets from embryonic stem cells, an endless source. And there are trials underway, again, including in Canada, with this approach. And then heart disease, one that is a harder target, I believe, is where the idea is to actually replace damaged heart cells. And there's some very exciting preclinical data, including right here in Toronto with Blue Rock Therapeutics. So it's happening, and it is coming down the road. So it's very exciting. But you shouldn't forget that again, pluripotent stem cells have the biggest potential for the broadest base of diseases. But there are stem cell therapies already in the clinic. It's not that we're waiting endlessly for pluripotent cells. Bone marrow transplantation, going right back to Till and McCullough, is of course a stem cell therapy. You're taking the cells from the bone marrow that contain the stem cells that Till and McCullough identified, and they are being used to treat cancer, treat immune deficiencies, and for various blood-borne diseases. And in fact, an increasing range of treatments where bone marrow transplant comes into play. Skin grafts for burn patients, that's a stem cell treatment. You take a small piece of skin that's not burnt, and you can expand it using the stem cells in the skin into grafts that can replace the surface area of burns. So these are just a couple of examples where already adult stem cells are being used in the clinic. And they usually use their own cells or in a tissue-matched donor. So I just want to throw this slide in because if I don't, somebody's going to ask me anyway, because I always get asked this. So I've told you that pluripotent stem cells come from either embryos or by reprogramming adult cells. I've told you that there are adult stem cells in the bone marrow, in the skin, and other places that can be used for therapy. Well, what about cord blood? Isn't cord blood a great source of everything? Well, it is true that umbilical cord blood is a good source of blood stem cells. Not other cell types. Umbilical cord blood stem cells are not pluripotent stem cells. Also, although they're a great source of cells, there's only enough cells from one cord, one baby, for one transplant to one small child. So the concept of putting away the cord blood from a baby to treat that baby when it grows up is really not appropriate. It's highly unlikely that that cord blood would be sufficient to treat the child. Also, the most usual treatment, the usual disease that's being treated by a transplant in a child is leukemia, and you would not put the child's own cord blood back in that situation because you'd worry about genetic changes there. So this is another case where what's important is having a public bank. So cord blood banking is a good idea, but it should be done in a public bank. So you put in the baby's cord and you pull out what is needed and matched at a later date. So cord blood has its uses. There's a further extension that people talk about, and you see it all the time, where there are, of course, stem cell clinics worldwide that are proliferating, selling unproven cell therapies that they claim to be stem cells and claim to treat many, many different diseases. Most of these are unproven, and unless there is clinical trial data, are really not something that should be trusted as a major source to treat disease. This is what we call stem cell tourism. And I'm going to show you a couple of slides I borrowed from my friend Tim Caulfield at the University of Alberta, who really has taken a very broad approach to this. And you can find him online, has many different talks and different lectures that he's given on this issue. But this is just one case where, this was actually a couple of years ago, he just, before giving his lecture, he said, oh, like, I'm going to go on Google and enter stem cell therapy. And you can see what the result was. 48 million results in 1 second. And they just go on and on and on. So this is one in Mexico. You can treat vasculitis, Crohn's disease, autism, Alzheimer's, paralysis, ALS, COPD. Here's another one that it will also treat erectile dysfunction with stem cells, male infertility, and so on and so forth. Not only clinical treatments, but of course, stem cell has become a good term that everybody feels must be good. If you have a stem cell in it, you're going to be young and rejuvenated. So there's a lot of these kind of things, stem cell cream, fountain of stem cell youth. This is one of Tim's articles. And you know, this product has been rigorously tested for maximum efficacy. Proteins from human non-embryonic stem cells, stemology, rejuvenating benefits of stem cells without all the ethical baggage. That's because they aren't stem cells. And you know, you can probably apparently you can get them from apple stem cells as well. So this is another very difficult area, and it is very important to be very cautious about claims of stem cell therapy clinics where we don't have scientific proof of efficacy. And there are lots of websites. Here's a couple where you can, if you have patients who have questions, you can go to these websites and get more information. So stem cell therapies, I honestly believe, are changing the future for serious degenerative diseases. But there are other kinds of diseases and other kinds of approaches that are also becoming accessible to new technologies. And I want to talk now, I'm going to switch to talk about serious genetic diseases and whether we can learn how to correct genetic diseases using gene therapy and gene editing. And this is where we come to the next game changer that I want to talk about, and that's not stem cells, but this is CRISPR-Cas9 gene editing. So a little bit of hardcore science for you now. For those of you who know the science, this is fine. For those of you who don't, hang in there. So this is what we call a gene editing system. And this is a little enzyme. This little blue thing in the middle is an enzyme. It's a nuclease. That is to say, it cuts nucleic acids and it cuts DNA. And it does that in a very specific manner, because what it does, you can take a piece of RNA that lines up to the DNA you want to cut, and the guide RNA will guide the nuclease to that specific site, and then the nuclease will cut it. That cut is so precise that you can take out a piece of DNA, you can put back in a piece of DNA. Modifications of these enzymes can change just one base pair to another base pair. You can insert large pieces of DNA. Essentially, I like to think of this, and this is the way most of the CRISPR people talk about it as well. It's an editing system, the same way like a word processor on your computer. You can edit the sequence of the DNA. Remember, you've only got 4 bases in DNA. 4, that's 5, 4 bases, and you can change those now at will. It's a system that bacteria use to cut and edit DNA. And actually, they use it to get rid of their own viruses. So when viruses invade bacteria, the bacteria uses this gene editing system to cut up the viruses and get rid of them. The system is extremely specific, extremely efficient, and importantly, cheap. The components here, there's nothing expensive here. This is very easy for anyone to do. And in the time since this was first discovered, which is not so very long ago now, this system has been used to make genetically modified plants, animals, bacteria, and fungi. So CRISPR-Cas9 gene editing is changing the way we, as a research tool, use this in the lab. So we can modify mice, we can modify cells. It's useful in agriculture to make modified agriculture, animals and plants. And it has the potential to be able to modify the human genome. I like this slide because, in fact, the discovery of the CRISPR enzymes came about from researchers at Danone who work on yogurt, because your yogurt has a very specific bacterial culture that's necessary to make the yogurt. If that culture gets infected by a virus, you're in trouble, and Danone's in trouble. So they had researchers trying to work out how bacteria fought off those viruses. And it's that work that essentially began the work with CRISPR. So again, you never know where science is going to come into play. So there is CRISPR in your yogurt. And some of the people who worked on CRISPR in yogurt are actually the 2 gentlemen at the bottom here, Philippe Horvath and Rodolphe Barangu. And along with the 3 people on the top, they shared the Gardner Award in 2016 for the discovery of the gene editing, CRISPR gene editing process. So they discovered it in bacteria. Emmanuel Charpentier and Jennifer Doudna turned it into a system that they understood the biochemistry and could use it in any other cell type, not just bacteria. And Feng Zhang at MIT really democratized this and made it into something that anyone could use around the world, which is really, really important and a great contribution to the community. And of course, you may remember that last year, Jennifer Doudna and Emmanuel Charpentier won the Nobel Prize in Chemistry for their work on the CRISPR-Cas9 system. So sometimes these game changers like iPS cells and CRISPR they get the Nobel Prize very quickly. Although think of the Nobel Prize being many, many years later, but in this case, they really did get there very quickly indeed. So we can edit the DNA. We can use it in research. I use it a lot to make genetically modified mice. We have lots of pretty mice where we have GFP under the control of different genes, and we can study developmental processes. But how are we going to apply it into human genetic disease? So this is what we call somatic gene editing. So a person who has a serious genetic disease, a single gene mutation, we should be able to, with genome editing, go into the right cells and fix that mutation, or perhaps add a gene to a specific site and get that expressed properly in those cells and modify the progress of the disease. So genome editing means that you can essentially treat serious single gene diseases by correcting mutations, by modifying the genes themselves, or inserting genes at defined sites. There are two different kinds of ways you could think about this. So we call it somatic, because somatic means everything except the germ cells, everything except the eggs and the sperm. And you'll realize why I'm saying that in a minute, because we're going to go on to talk about what happens if you make genetic changes in the eggs and sperm. So this would be for people who have a genetic disease and you want to treat them. So the idea is that you go into those people and fix the gene. There are two possible ways of doing it, depending on the tissue that's being affected by the genetic change. You can do what we call ex vivo. So you take out the cells, edit them, and put them back. This is mostly applicable to blood-borne diseases. Back to our bone marrow transplants again, take out the cells, do the genetic trickery, and put them back. Importantly, because CRISPR-Cas9 editing is so efficient, it's possible now to think about doing this kind of genetic alteration directly in the body, taking the components of the gene editing system, putting them in a virus or in a nanoparticle. Sound familiar from messenger RNA vaccines? Putting them into a virus or into a nanoparticle and taking that and put it back directly in the body and actually genetically modify the cells right there in the person. So this is really amazing. And this is moving very fast to clinical trials. Here, this is 2016, listing some of the potential targets for therapeutic genome editing. They include a number of genetic eye diseases, Leber's congenital amaurosis. There is already a gene therapy trial underway for that disease. Retinitis pigmentosa is next in line. Liver, a number of diseases, including hemophilia. There are beginning to be the first phase 1 trials of going directly into people and putting viruses with the CRISPR components into those people, infecting the liver and getting hemophiliac patients to produce the proper factor VIII to prevent hemophilia. Skin, there's been a beautiful study already with kids with epidermolysis bullosa, which is a blistering disease of the skin. They lose large amounts of their skin. Using skin stem cells, which I already described, you can genetically modify, fix the mutation from the kid's own cells, and then expand those and use them to repair the damaged tissue. It's amazing, and it's happening. Lung cystic fibrosis. There's no trials yet, but this is an obvious target where you could go in and actually fix the defective gene, which we've known for a long time, and fix the gene in the lungs. Blood, all of these are actually underway right now. Sickle cell disease and beta thalassemia, there are trials and first patient results suggesting using gene editing that you can actually really help get over the problems with sickle cell disease and beta thalassemia by modifying the patient's own cells. And this is the one I always like to just throw up at the end, because to me, this was the one that I thought would be absolutely impossible. So you all know, I'm sure, that muscular dystrophy is a terrible degenerative disease of the muscles, and it's caused by a mutation in a gene called dystrophin. It's a very large gene. It's X-linked, so boys have this disease, almost completely restricted to boys, and it's progressive and fatal. And every muscle in the body, including the skeletal muscle and the heart, are affected by this mutation. So how could you possibly fix it? So how could you actually go in and get enough repair, enough gene editing to cause some alleviation of the symptoms? So, well, this is not humans, but this is dogs. Mice first, but then dogs, which is a much larger animal. Work, particularly from Eric Olson's lab in Houston, have been able to show, and I'm not going to go through the details here, that they were able, using, again, a virus carrying the gene editing components, they could take this dog, where this is the expression in the wild type dog. In the muscle, this green is the dystrophin protein. This is the mutant, no dystrophin to speak of at all. When they express the gene editing tool and fix the mutation, now you see not every cell, but lots of cells are now expressing dystrophin, and they were able to show that they got some recovery of muscle strength in these animals. It's really incredible, and this is moving very rapidly towards clinical trials. So gene editing for serious genetic diseases, single gene diseases, is transformational, and you're going to see that in the clinic now and in the next few years. But there's always a but. Let's get to the ethical concern. Why has gene editing become controversial? Not so much, I don't think, for treating people who have serious genetic diseases. Why would you not want to treat people with disease by somatic gene therapy? But the question is, well, why, if we can fix genes like that, why would we wait to treat a person who has a disease? Why don't we just prevent the disease by modifying the gene right back in the early embryo and make a baby that doesn't have that mutation? Wouldn't that be a good idea? So that's what we call heritable genome editing. So somatic is treating the tissues of the person who has the disease, and it doesn't change their offspring. If they carry a genetic mutation, their offspring will carry the genetic mutation. Heritable gene editing, the idea is to actually prevent transmission of that mutation into the next generation. And because of all the advances I've just told you with CRISPR and the efficiency, and indeed some of the techniques that my lab and others have shown work in the mouse, we know that it's a realistic possibility to think about modifying the DNA in the early embryo. And in theory, this would allow individuals to have genetically related children without passing on the risk of a genetic disease, could prevent genetic disease. But it has other, then the question is, where do you draw the line? Because it also has the potential to think about altering predisposition to diseases. So not single gene serious diseases, but other diseases where we do know some of the genes that predispose to cancer, to heart disease, to Alzheimer's. Maybe we should modify those genes in the early embryo. And then if you go further, well, if we're going that far, why don't we just think about enhancing humans and making better humans? So this is where we get into this concept of designer babies. So you can find many images like this on the internet. You know, could we make a baby with high IQ, 20/20 vision, perfect pitch, sprinter? I don't think we know enough about IQ to be able to do this. We do know quite a bit about muscle development. We could probably modify genes and make a better sprinter. But what I put on this slide is, of course, what you could do is essentially cure this kid of cystic fibrosis or Huntington's disease, serious genetic diseases, by essentially preventing that in the embryo itself. Now, it's true to say that almost all these single gene diseases, there are ways of preventing the transmission that don't require gene editing, which we call pre-implantation genetic diagnosis. So instead of modifying the embryo, you take a sample of that embryo and basically pick the embryos that don't carry the genetic mutation. And we can talk about that. You can ask me questions about that later. But essentially, for most situations, that's going to be perfectly acceptable. However, there are situations where it is not possible to think about picking out the right embryo that doesn't have the mutation, and it would be nice to be able to fix it. But the question is, where do you draw the line? Where do you draw the line between preventing a serious genetic disease, altering the predisposition to serious disease or enhancing human development. Where do you draw the line? And this has been many working groups have been discussing this. It seems to me endlessly. I was on this working group from the National Academies in the US. It also include the Royal Society in London and the Chinese Academy of Sciences. We had a big international group. We spent a year discussing the whole issues around human genome editing, somatic and heritable, and we had a lot of stakeholder engagement. And I will summarize the whole discussions in this sort of traffic light image here. The major recommendations of that report in 2017, and most of the other working groups that have taken place around the world can be summarized as this: green, yellow, and red. Green is okay to use genome editing as a tool. It's an incredible tool for basic research, understanding gene function, including in human embryos in culture, but not in embryos to be put back for pregnancy. Somatic gene editing to treat disease, I've just told you how exciting that is. And certainly there's a green light there. We need to be able to understand it. We need good clinical trials. We need to have safety concerns assessed, but they're no different from other forms of gene therapy. Yellow is how this group came out for germline editing. And yellow means we're not there yet. We don't think it's safe enough. We don't know enough about the process. We don't know what the long-term outcomes would be. But if those can be addressed, then germline editing could be used to treat disease where there's no alternative and the safety issues are addressed. So very serious genetic disease for which there is no alternative. And red, no way should we be using somatic or germline editing for enhancement purposes. So this is fine, and working groups come out and everybody agrees, but they come out as working groups and they don't really have any backup. There's no legislation, no regulation behind this. This is all, these are good things and this is what we should do. So what happened, of course, is that somebody somewhere is going to go ahead and in 2019, Dr. He in China claimed to produce gene-edited babies. And what he did was to produce twins in which he'd used CRISPR-Cas9 to modify a gene, CCR5, which if you knock it out, though you prevent HIV infectivity. So patients who have mutations in this gene are resistant to HIV. That, of course, does not fall, if we go back, that there are plenty of alternatives for treating HIV, and going as far as mutating the receptor is not one of the recommended alternatives. So there was no reason for him to do that. It also, of course, was not safe because we do not have enough information at this point about off-target effects and other effects in the early embryo. So this caused a big uproar, and he didn't do very well out of it. The Chinese government, the Chinese academy, his university, everybody came down on him like a ton of bricks. And apparently he was sentenced to 3 years in prison for illegal medical practices. I believe he's out of prison now, but I do not know where he is or what he's doing. And importantly, I don't know what happened to the babies. So this started another round of discussions. And there's been 2 big working groups that came out last year. This is another National Academies working group. I was not on it, but I was on the oversight panel for this. And this NAS report basically looked in more detail at what it would take to get heritable gene editing into the clinic, and still came out basically with the same, I think, the same conclusions that we came to in 2017, that it's still too risky. The safety and efficacy of genome editing in human embryos has not been proven, researchers warn. A second study from the from WHO also came out looking at how to really set up an oversight and a way to at least have a database of what's going on in human gene editing worldwide. And they're proposing that essentially there should be a kind of biobank, if you like, of experimentation, so everybody knows what's going on. I think that's a nice idea, but I think WHO has several other things on its mandate right now, and this is not one that's going to get a lot of attention. But it is still important. I've made it sound as though, you know, we're going round and round in circles, and I would say we are. There's a general consensus in the scientific community that we are not anywhere near ready for heritable gene editing. And as I say, it's not altogether clear that there are very many situations where it's the right choice to go forward anyway. Somatic gene editing has huge potential, and you don't want to see that being stopped because people are concerned about the heritable side of things. So that's the sort of run through. I hope I've taken you from the embryo to stem cells to gene editing. We've gone from mice to humans. We've gone from stem cells to embryos. We've gone from gene therapy to design of babies. And it's all extremely exciting. And it's an area of science where it's moving extremely fast. There are lots of other areas related to all of this that are going on today that I could spend another lecture talking about. But the fact is, it's moving extremely fast. There's a lot of exciting information. We're learning more about human development. We're learning more about stem cells. We're developing clinical treatments for degenerative diseases, and we're developing, it's a royal we, the field is developing gene editing tools to really treat serious diseases. But in all of these cases, and some of the other areas as well, there are big ethical, legal, and public policy implications, and one can't ignore that. And as scientists working in the field, we obviously have to be engaged with that. But it's not enough, of course, to have the scientists. We really need to engage a broad range of stakeholders, including patients, public, policymakers, to debate how and when and where to draw the boundaries. So I will stop there. Thank you. And